So today we'll talk about a bit of immunology, and we'll start with talking about the major histocompatibility complex, which is the topic for today. We'll see how is it made, how it processes antigen. Now, first you should know one important thing, that these MHC molecules were the ones which were historically known to cause transplant rejections or graft rejections, right? Because they were the ones who were supposed to make and process and present such antigens which would lead to transplant rejection. Now it has been known that it has very many functions in immunological responses. Now, before we go into the details of MHC processing, first we should see some major differences between the classes of MHC proteins which are present in the human body. Now, remember that in the human body, these MHC cluster, which is present in particular genetic loci, they are known as human leukocyte antigen, right? So these human leukocyte antigen, they are regions in the genetic loci which are actually encoding for these MHC proteins. Now, starting with the differences, so just put on the heading here as MHC1, MHC class 1. Then here we have MHC class 2, right? So the first difference you should know is in the genetic loci. As we said, that the genetic loci is actually in humans known as HLA. Now, human leukocyte antigen, this was the full form of HLA, and here MHC class 1 protein is encoded by the genetic loci HLA-A, HLA-B, and HLA-C region, whereas MHC class 2 is encoded by HLA-D, and that too the subunits are DP, DQ, and DR are the subregions, right? Now, the next point of difference is the composition. What do you think are MHC molecule composed of? Obviously, they are MHC proteins, right? So they are composed of polypeptides. Now, the main thing that you should remember, what kind of subunits MHC1 has. MHC1 has a transmembrane glycoprotein. Okay, I'm just writing here it as transmembrane. Okay, just write it here. Glycoprotein. Along with that, it has a beta-2 microglobulin. But this beta-2 microglobulin, remember, it is not encoded by the MHC region. Okay, not encoded by the MHC region. Now, what is the composition of MHC2? MHC2 has an alpha chain, a beta chain, and an invariant chain, which I'll demonstrate it by Li. Now, the alpha chain, if you should know, it is of 33,000 Dalton. That is the molecular weight. 29,000 is of the beta chain, and the invariant chain is of 30,000 Dalton. These are the molecular weights I'm talking about, right? Transmembrane glycoprotein, remember, it has a molecular weight of 45,000 Daltons, whereas the beta-2 microglobulin has 12,000 Daltons. Right. Now, apart from the MHC1 protein and MHC2 protein composition, there are other features of differentiation. What do you think? Which kind of cells they are present in? So, cell distribution we're talking about. So, which kind of cell they are present in? The first kind of cell, actually MHC1 is present in all types of cells, all somatic cells. There are two exceptions to it. All somatic cells except the brain cells and retinal cells. Okay. Now, in the brain and the retina, you won't have an MHC class 1, whereas all somatic cells will be present with MHC class 1 protein. Now, MHC class 2 proteins are present in specifically, it has a specificity towards the cell distribution, and they are mostly present with the B cells, right? The macrophages, as well as the dendritic cells. Or what you can write for all these three are the antigen presenting cells. That is what it is known as APCs. So this is where they are mostly present, right? They have the specificity to be present in these particular cells. Now, after the cell distribution, what one should know is about the activation. Which system or which hand of the adaptive immunity is these MHC class proteins activating? MHC class 1 activates the CD8 T cells, right? Whereas MHC class 2 activates the CD4 T cells. Okay. Now, apart from activation, we should also know, just let me scroll it down. Apart from activation, we should also know that how many residues of antigen the MHC class can bind to. So MHC1 actually binds to 8 to 10 residue. The reason is, and this binds to 10 to 30 or more than that residues. Now, the reason is because MHC class 1 protein has a structure or has a folding due to which there are many constraints for the antigen to come and bind. Okay, it's a very small space for the antigens to come and bind. That is why the residues number is very less. That is why 8 to 10 residues are actually binding to the MHC class 1 protein, whereas MHC class 2 is naturally having a larger space to bind. That is why around 10 to 30 residues of antigen can actually bind to MHC class 2. Okay, so now let's talk about the antigen processing, how it takes place. Now, first, let me just mark this cell as the APC. This is the antigen presenting cell. It can be any of the antigen presenting cell. It can be macrophages, it can be B cells, or it can be the dendritic cells, right? Any of these antigen presenting cells, as their name suggests, what is their function? That they are going to take the exogenous or the foreign antigens which are not endogenous to ourselves, and they are going to present it to the adaptive arm of the immunity, right, which is the cell mediated immunity. It is going to be presenting it to these foreign antigens to the T cells, right, so as to initiate the immune response. Now, it can be done in two manners. One is the class 2 MHC pathway and the other one is the class 1 MHC pathway. So let's start with the class 2 MHC pathway. As you can see that this is the rough endoplasmic reticulum. Class 2 MHC is actually a protein. Just now we studied in the table, it has three compositions: the alpha chain, the beta chain, and the invariant chain. As you know that the invariant chain is responsible for what? It is going to bind to the alpha and beta chain. Why? Because it does not want, or it prevents the endogenous peptides, these endogenous peptides, to actually bind to the alpha and beta chain, right? So this MHC2 protein as a whole, if you see, it consists of this invariant chain, which is responsible for preventing the binding of the endogenous peptides. Why we are preventing the binding of endogenous peptides? Because we want that the exogenous peptides or the foreign antigens, they are the ones who are supposed to bind, and they are the ones who are supposed to come inside and bind, and not these endogenous peptides. So to prevent the binding of this, the invariant chain is bound to the alpha beta chain. Now, this complex is together known as it goes into the Golgi apparatus and into the exocytic vesicle, it is together known as MHC2 compartment. This is the MHC2 compartment. Right. Now, as you can see, the exocytic vesicle, it now fuses with another vesicle. Now, where is this another vesicle coming from? Just look below here. This is the exogenous antigen. This may come from any kind of bacteria. Now, this exogenous antigen, when it is exposed to this antigen presenting cell, what does any, for example, macrophage do? It is going to internalize this antigen, right? And then send it to the endosomes, where these antigens are going to be either denatured into its primary state or proteolized, right? Partial proteolysis will occur, and these peptide fragments are going to be formed. And these peptide fragments, what are they going to do? They are going to then be fused with the alpha and beta chain of the MHC class 2 protein. Now, what happens to the invariant chain? It has to be detached and it has to expose the sites where these peptide antigen can fit inside it, right? Now, once the peptide antigen, you can see this green color molecule is the peptide antigen, and this square, which was earlier what? The alpha beta chain, right? This. Now, they all together are going to be exocytosed from the cell and going to be present on the cell surface. You can see they are now presented on the cell surface, and now this is known as together peptide antigen MHC2 complex. Surface peptide antigen MHC2 complex. This is what they are known as. Now, this is the MHC class 2 pathway. Now, what about the MHC class 1 pathway? What happens in MHC class 1? It is directed more towards a virus infected cell. For example, all these macrophages, dendritic cells, and the B cells, these antigen presenting cells, are already infected by a virus. So what happens? Virus comes inside and it takes up the host cell machinery and starts synthesizing its own proteins. Now, because of this, viral proteins are synthesized, and viral proteins will now go inside this proteasome. Now, once it reaches the proteasome, what happens is this proteasome is going to cause proteolysis of the viral proteins, because of which these endogenous peptides. Now you remember that why I told here this endogenous peptide was prevented to be binding to the MHC class 2. That is because the endogenous peptides are then known when already the virus is coming inside, forming its protein inside the cell. Now that is why these peptides will be known as endogenous peptide. And now these are the ones which can actually go and bind to the MHC class 1 protein with the help of what? With the help of transporter associated with antigen processing, TAP. Now this TAP is what? Transporter associated with antigen processing. This is a transporter located on the rough endoplasmic reticulum, and these endogenous peptides which are formed is going to be transported with the help of this transporter into the rough endoplasmic reticulum, and with the help of this alpha beta 2 microglobulin. These were the two composition of the MHC class 1, this transmembrane glycoprotein and the beta 2 microglobulin, right? And now it will bind to these two structure, it can form the complex, and now with the help of Golgi body and again exocytosis, it is going to be presented on the cell surface. So this is how antigen class 1 MHC complex is going to be formed and be presented on the cell surface. Now, what will happen once they are presented on the cell surface? Where are they going to go? In the table we had written down, if you remember, that they are going to actually be formed and be presented to the T cells, right? MHC class 1 is going to be formed and presented to the CD8 T cells, whereas MHC class 2 is going to be presented and that will activate the CD4 T cells. Now, as you can see in this picture, I would like to highlight the point. Now, this was the antigen presenting cell, the MHC, this is the cell membrane of it, cell surface, and here the MHC protein is actually presenting the peptide fragment of the antigen, and now it is telling this T cell to get activated because this peptide antigen is not of self, it is foreign antigen, right? So that is what MHC is responsible to do. That is the main function. That means it is going to differentiate between self and non-self. That is why when we say that some people have more immunity and some have less towards a specific pathogen, that is when we say that because of various alleles or various genetic alleles of the MHC protein, some people have the ability to confer a stronger immune response than the other, because each of our MHC allele differs from one another. The entire population MHC allele will differ from one another, and this pleomorphism will help to confer, some will be able to confer higher immunity than the others, right? This is one area of differentiation. Okay. Now, as you can see that this is the peptide antigen which is presented to this T cell. When we do a video on T cell, you will know that this is the T cell receptor and what all subunits are present, we will study it there. Now, in contrast, you can see that here there is no peptide antigen. But this super antigen, what is this now? Now, this super antigen is actually binding to the class 2 MHC molecules, but outside the peptide. This is the cleft formed by the MHC protein for the binding of peptide antigen. But outside this cleft for the peptide antigen is binding the super antigen. So some points of differentiation between super antigen and peptide antigen is that super antigen is basically the antigen which is going to elicit a higher, at least 25% more immune response than a normal peptide antigen, right? It is a much larger antigenic molecule than the peptide antigen. Okay. The other point of differentiation is that this super antigen is binding to the, as we've already said, it's binding to outside the peptide binding cleft, right? Okay, so let's write down some example for the super antigen. One example we all would know is the staphylococcal. Staphylococcal. Okay, one second. Okay, staphylococcal enterotoxin. Staphylococcal enterotoxin. Now, there is another TSST1, which is toxic shock syndrome toxin 1, right? This enterotoxin is the one which causes food poisoning, right? Then there is also group A streptococcal pyrogenic exotoxin, right? So exotoxin A. So these two can be the examples for super antigens. And what does a super antigen do? We know that it is going to elicit lots of immune response, at least more than 25% of X. If peptide antigen is going to elicit X%, this is going to elicit more than 25% of X. Now, one more thing, remember, as the super antigen is going to elicit the immune response, it is going to activate interferon gamma. Okay, interferon gamma, and then interferon gamma is going to activate the macrophages, and there is going to be interleukin 1, 6, and TNF alpha, which is going to be produced. And this is actually known as what? This is known as the cytokine storm. It is a storm of cytokines which are actually being elicited because of the super antigen. And this may lead to like some complications like organ failure, shock, hypotension, right? So this was all about MHC classes of proteins and MHC processing and antigen presentation.